talk about protein structure. Understanding the structure of proteins is a vital part of understanding pharmacology. Remember, proteins make up a large number of drug targets, both in the human body and in microorganisms. Proteins are pretty complex, and there are a lot of details that we won't review for now, but their structure can be broken down into 4 basic levels of organization: primary, secondary, tertiary, and quaternary. Let's spend a few minutes refreshing our minds and discussing these so that when we see crazy, complicated looking images like this, we know what we're actually looking at. So first, naturally, we have the primary structure. The smallest building block, or monomer, of a protein is an amino acid. Generally, we are most concerned with the 20 amino acids typically used in human protein synthesis. I've linked an excellent video in the description to help you memorize these if that's something you're interested in. The primary structure of a protein is the sequence of amino acids. Technically speaking, a string of 2 to 50 amino acids is referred to as a peptide. Peptides are further divided into different classifications depending on the number of amino acids in the string. When you get to the 50 mark, the peptide is considered a polypeptide. Anything above 50 is generally considered to be a protein. So it makes sense that when we are looking at a protein, especially bigger proteins with longer strings of amino acids, we are looking at multiple polypeptides strung together, also known as a polypeptide chain. So now we have our primary structure, our long string of amino acids. Inevitably, as these linked amino acids start to brush up against other parts of the string, intermolecular forces will start to do their thing. It turns out that when this happens in localized regions of the polypeptide chain, it tends to follow certain patterns. These patterns make up the secondary structure. First, we might see something called a beta-pleated sheet. These always make me think of Ruffles chips. We might also encounter a soft-serve ice cream looking spiral called an alpha helix. There are also beta barrels, which are too crazy for me to try to draw, but I've included a picture so that you would have a general idea of what these look like. The intermolecular force that is primarily responsible for patterns that make up the secondary structure of a protein is hydrogen bonding. A protein's tertiary structure is what happens when sections of our polypeptide chain start to interact with other sections further along the string. Remember, we still have our secondary structural components like our alpha helices and our beta-pleated sheets. Once again, we have hydrogen bonding playing a role in our tertiary structure, but we also start to see other types of interactions like covalent disulfide linkages. What we are left with at this point is a complete protein subunit. In a process called oligomerization, multiple subunits come together, forming the quaternary structure of our protein and giving us our final product. So, in review, amino acids are organized into a specific sequence known as the polypeptide chain. Then, amino acids in one part of the chain interact with other nearby segments of the chain via hydrogen bonding, forming the components of our secondary structures such as beta sheets, alpha helices, and beta barrels. Our tertiary level of organization happens when segments of the polypeptide chain interact with other segments that are further away through hydrogen bonding and covalent disulfide linkages. This ultimately forms our complete protein subunit. Finally, multiple subunits come together, giving us the quaternary organization of our protein. And that's it. I hope that this helped you wrap your mind around how proteins are organized. If you enjoyed this content, please like and subscribe. Stay curious, and I'll see you in the next video.